Assalamualaikum warahmatullahi wabarakatuh. Good morning. I hope that all of you who are watching are in good health. Amin. Here we are a group consisting of myself, Muhammad Zaldi, Ahza Nadhif, and Gladys Gwendolyn. We want to present about the cell structure and function of the organelles, endosymbiont theory, prokaryote structure. The cell structure. The structural cells existing in animals and plants. And that is the structure. That is the structure. The first is ribosomes. Ribosomes are small cell organelles that are about 20 nanometer in diameter. Ribosomes are found in the cytoplasm, are attached to the AR membrane during the synthesis process. And then lysosomes. Lysosomes are cell organelles that are small, slightly rounded sacs, and are bound by a single membrane system. And then nucleus. The nucleus is the large cell organelles and contains genetic information in the form of DNA. The nucleus is a cell organelle that is very important for life because it controls all cell activities. And then nucleus, a membrane-bound structure consisting of proteins and nucleic acids in the nucleus of a cell. And then mitochondrion. Mitochondria are cells organelles that have different shapes. Some are oval, round, cylindrical, and irregular. And then rogue endoplasmic reticulum. Rogue endoplasmic reticulum, RER, series of flattened part of continuous membrane organelle within the cytoplasm of eukaryotic cell. That is plays a central role in the synthesis of proteins. And then smooth endoplasmic reticulum. Smooth endoplasmic reticulum, SER, a network of smooth disc-like tubular membrane vesicle, part of a continuous membrane organelle in the cytoplasm of eukaryotic cells, even involved in the synthesis and storage of lipids, including cholesterol and phospholipids, which are used in the production of new cell membranes. Vacuole. The vacuole is a cavity that forms inside the cells and is lined by the tonoplast membrane. In the plants, vacuoles are large, as in parenchyma and collenchyma cells. Peroxisome. The peroxisome is an organelle enclosed by a single membrane of lipid bilayer containing absorbing proteins receptors. Peroxisomes don't have a genome and contains about 50 enzymes such as catalase and urate oxidase that crystallize in the center. Peroxisomes are found in all eukaryotic cells. And then Golgi apparatus. Golgi bodies are organelles that form complex mesh-like structure. and then cell membrane. The cell membrane, also called the plasma membrane, is found in all cells and characterizes the interior of the cell. From the outside environment, the cell membrane consists of a lipid bilayer that is semi-permeable and last, cytoplasm. Cytoplasm is the part of the cell that is enclosed by the cell membrane. In eukaryotic cells, the cytoplasm is the non-nuclear part of the protoplasm. In the cytoplasm, there is a cytoskeleton, various organelles and cycles, and a cytosol, which is a liquid in which the organelle float around it.
Cell organelles and function. Cell consists of some organelles. Some of them are cell membrane, cytoplasm, mitochondria, nucleus, ribosome, lysosome, endoplasmic reticulum, Golgi apparatus, vesicle, cell wall, plasmodesmata, chloroplast, and vacuole. First one is cell membrane. Cell membrane, also called the plasma membrane, is found in all cells and separates the interior of the cell from the outside environment. The cell membrane consists of lipid bilayer that is semi-permeable. The cell membrane regulates the transport of materials entering and exiting the cell. The cell membrane is present in both plant and animal cells. Cytoplasm is a thick solution that fills the cell and is enclosed by the cell membrane. It is mainly composed of water cells and proteins. It is the medium for chemical reaction. It provides a platform upon which other organelles can operate within the cell. All of the functions for cell expansion, growth, and replication are carried out in the cytoplasm of a cell. Mitochondria are often referred to as the powerhouses of the cell. Mitochondria are cell organelles that have different shapes. Some are oval, round, cylindrical, and irregular. They help turn the energy we take from food into energy that the cell can use. Mitochondria are also involved in other tasks, such as signaling between cell and cell death, otherwise known as apoptosis. The nucleus is the largest cell organelle and contains genetic information in the form of DNA. The nucleus is a cell organelle that is very important for life because it controls all cell activities, and the nucleus is the site of transcription. Transcription creates different types of RNA from DNA. Nucleus is found only in eukaryotes and is the defining characteristic feature of eukaryotic cells. A ribosome is a complex molecular machine found inside the living cells that produce proteins from amino acids during a process called protein synthesis or translation. The process of protein synthesis is a primary function which is performed by all living cells. Ribosomes are specialized cell organelles and are found in both prokaryotic and eukaryotic cells. Every living cell requires ribosomes for the production of proteins. A lysosome is a membrane-bound cell organelle that contains digestive enzymes. Lysosomes are involved with various cell processes. They break down excess or worn-out cell parts. They may be used to destroy invading viruses and bacteria. If the cell is damaged beyond repair, lysosomes can help it to self-destruct in a process called programmed cell death or apoptosis. A continuous membrane system that forms a series of pattern sacs within the cytoplasm of eukaryotic cells and serves multiple functions, being important particularly in the synthesis, folding, modification, and the transport of the proteins is endoplasmic reticulum. It is divided into smooth and rough ER. Smooth ER doesn't have ribosomes. It produces lipid and steroids. Rough ER has ribosomes. It synthesizes proteins. Golgi apparatus, also called Golgi complex or Golgi body, membrane-bound organelle of eukaryotic cells that is made up of series of flattened-stack pouches called cisternae. The Golgi apparatus is responsible for transporting, modifying, and packaging proteins and lipids into vesicles for delivery to targeted destinations. It is located next to the endoplasmic reticulum and near the cell nucleus. While many types of cell contains only one or several Golgi apparatus, plant cells can contain hundreds. In cell biology, a vesicle is a structure within or outside a cell consisting of liquid or cytoplasm enclosed by a lipid bilayer. Vesicles form naturally during the process of secretion or exocytosis, uptake, or endocytosis and transport of the material within the plasma membrane. Vesicles are involved in metabolism, transfer, buoyancy control, and temporary storage of food and enzymes. They can also act as chemical reaction chambers. The cell wall is a structural layer surrounding some types of cell just outside the cell membrane. It can be tough, flexible, and sometimes rigid. It provides the cell with both structural support and protection, and also acts as a filtering mechanism. Cell walls are absent in animals, but are present in most other eukaryotes, includes algae, fungi, and plants, and in most prokaryotes except molecular bacteria. A major function is to act as pressure vessels, preventing overextension of the cell when water enters. Plasmodesmata are small channels that directly connect the cytoplasm of neighboring plant cells to each other, establishing living bridges between cells, similar to the gap junction found in animal cells. The plasmodesmata, which penetrate both the primary and secondary cell walls, allow certain molecules to pass directly from one cell to another, and are important in cellular communication. Chloroplast is an organelle that contains the photosynthetic pigment chlorophyll that captures sunlight and converts it to useful energy, thereby releasing oxygen from water. Chloroplasts are found in all green plants and algae. They are the food producers of plants. These are found in mesophyll cells located in the leaves of the plants. They contain a high concentration of chlorophyll that traps sunlight. This cell organelle is not present in animal cells.
We will talk about prokaryote structure. Prokaryotes are some of the oldest life forms on our planet. They have no nucleus and show huge variation. Many people know them better as bacteria, but although all bacteria are prokaryotes, not all prokaryotes are bacteria. Prokaryotic cells are generally 1,000 times smaller in volume and lack a nucleus that is surrounded by a double membrane. Prokaryotes are thought to have been the first living organisms on Earth. Most biologists believe that eukaryotes evolved from prokaryotes about 2 billion years ago. There are two groups of prokaryotes known as bacteria and archaea. Prokaryotes and eukaryotes difference in structure. Eukaryotes and prokaryotes share many features such as ribosomes, cytoplasm, cell membrane, flagellum, and cell wall. But there are many key differences. Prokaryotes have no membrane-bound organelles, and eukaryotes have no capsule, and their cell walls are structured differently. These are the structures of prokaryotes. There are some structures that sometimes present and some structures that always present. The structures that always present are cell wall, cell surface membrane, cytoplasm, circular DNA, and ribosomes. Structures that sometimes present are flagellum, capsule, enfolding of cell surface membrane, plasmid, and pili. First, capsule. The capsule is a protective layer possessed by some bacteria that enhances their pathogenicity. The surface layer is made up of long strings of polysaccharides, long chains of sugar. Depending on how well this layer is stuck to the membrane, it is called either a capsule or, if not well adhered, a slime layer. This layer enhances pathogenicity by acting as an invisibility cloak. It hides the cell surface antigens that white blood cells recognize. Both help to protect the bacterium from drying out and may have other protective functions. For example, a capsule helps protect some bacteria from antibiotics. Some capsules prevent white blood cells known as phagocytes from engulfing disease-causing bacteria. Next, the yellow part is cell wall. Bacterial cell walls contain a strengthening material called peptidoglycan, a sugar protein molecule. The cell wall protects the bacterium and is essential for its survival. It prevents the cell from swelling up and bursting if water enters the cell by osmosis. The precise makeup of this varies hugely from species to species and forms the basis of prokaryotic species identification. This organelle provides structural support, protection from phagocytosis and desiccation, and comes in two categories, gram-positive and gram-negative. Gram-positive cells retain the purple-green stain because their cell wall structure is thick and complex enough to trap the stain. Gram-negative cells lose this stain because the wall is much more thin. A diagrammatic representation of each type of cell wall is given opposite. Next, plasma membrane. Like all cells, bacterial cells are surrounded by a cell surface membrane. Next, the blue part is cytoplasm. The cytoplasm does not contain any double membrane-bound organelles, such as mitochondria. The cytoplasm plays, if possible, an even more important role in prokaryotes than it does in eukaryotes. It is the site of all chemical reaction and processes that take place in the prokaryotic cell. Next, ribosomes. Bacterial ribosomes are 70S ribosomes, slightly smaller than the 80S ribosomes of eukaryotes. As at most simple, the ribosomes of bacteria are smaller, made of different subunits than those of eukaryotic cells. As such, antibiotics can be designed to target prokaryotic ribosomes whilst leaving the eukaryotic cells unharmed. With no functioning ribosomes, the cell cannot complete protein synthesis. Why is this important? Proteins are involved in almost all cellular functions. If proteins cannot be synthesized, the cell cannot survive. Unlike in eukaryotic cells, ribosomes in prokaryotes are never found bound to other organelles. Next are pili. Pili are smaller, hair-like projections that sprout over the surface of most bacteria. These often act as anchors, securing the bacterium to a rock, intestinal tract, tooth, or skin. Without such structures, the cell loses virulence as it cannot hold onto the host structures. Next, flagellum. A flagellum is a hair-like appendage that protrudes from certain plant and animal sperm cells and from a wide range of microorganisms to provide motility. Some bacteria are able to swim because they have one or more flagella. Bacterial flagella have a much simpler structure than eukaryotic flagella. The bacterial flagellum is a simple hollow cylinder made of identical protein molecules. It is a rigid structure, so it does not bend, unlike the flagella in eukaryotes. It is a wave-shaped and works by rotating at its base like a propeller to push the bacterium through its liquid environment. As a result, the bacterium moves forward with a corkscrew-shaped motion. Last is nucleoid or circular DNA. The DNA molecule in bacteria is circular. It is found in a region called the nucleoid, which also contains proteins and small amounts of RNA. It is not surrounded by a double membrane, unlike the nucleus of eukaryotes. There may be more than one copy of the DNA molecule in a given cell. Next, endosymbiont theory. The mitochondrion and the chloroplast are both organelles that were once free-living cells. They were prokaryotes that ended up inside of other cells or host cells. They may have joined the other cell by being eaten, a process called phagocytosis, or perhaps they were parasites of that host cell. Rather than being digested by or killing the host cell, the inner cell survived and together they thrived. It's kind of like a landlord and a tenant. The host cell provides a comfortable, safe place to live, and the organelle pays rent by making energy that the host cell can use. This happened a long time ago, and over time, the organelle and the host cell have evolved together. Now, one could not exist without the other. Today, they function as a single organism, but we can still find evidence of the free-living past of the organelles if we look closely. In the 1960s, it was discovered that mitochondria and chloroplasts contain ribosomes which are slightly smaller than those in the cytoplasm and are the same size as those found in bacteria. Cytoplasmic ribosomes are 80S, while those of bacteria, mitochondria, and chloroplasts are 70S. It was also discovered in the 1960s that mitochondria and chloroplasts contain small circular DNA molecules, also like those found in bacteria. It was later proved that mitochondria and chloroplasts are in effect ancient bacteria which now live inside the larger cells of animals and plants. The DNA and ribosomes of mitochondria and chloroplasts are still active and responsible for the coding and synthesis of certain vital proteins, but mitochondria and chloroplasts can no longer live independently. Mitochondrial ribosomes are just visible as tiny dark orange dots in the mitochondrial matrix. This is known as the endosymbiont theory. Endo means inside, and the symbiont is an organism which lives in a mutually beneficial relationship with another organism.